Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. We are grateful to you for being such fantastic listeners. Thank you for your continuous support. So far, we've launched 129 episodes since February 2020. Let's revisit a previous interview while we record new podcast episodes with your favorite GPCR scientists. Before we dive into this one, we would like to take a moment and thank our Dr. GPCR Ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Did you hear? We are working hard to bring you the Dr. GPCR University. It's a program where you'll be able to take a course on your favorite topic or even create a course to share with the community. For that matter, we are looking for experts to create courses on anything GPCR related and can't wait to unveil these to the Dr. GPCR community. Enter your course and you'll get a chance to win a cash prize for the most voted course in the ecosystem. Stay tuned and keep an eye out for our waitlist in the Dr. GPCR monthly newsletter. Become a Dr. GPCR Ecosystem Premium member and enjoy rewatching all the Dr. GPCR Summit and Symposia talks, the video cast of our podcast, and soon take a course at the Dr. GPCR University. You can also message members privately and network with our community from all over the world. And now, let's revisit this fantastic episode. Hello, everyone. This is Yamina from the Dr. GPCR podcast. I hope everyone is well. We're getting closer to the holidays. It's December 2020. And I have the pleasure of having with me Dr. Alexander Hauser. We have been trying to get this episode on the books, but it finally happened. Hi, Alex. How are you?
Hi, Yamina. Thanks for being here.
Absolutely. Thanks for being here. And it's kind of early here in Boston, but I'm glad that we were able to get together today. So how was the weather in Copenhagen?
It's dreadful. It's this gray cloud here over the city. I think I haven't seen the sun in the whole of December. So on top of it, it's when you see the clouds, it's gray, but then on top of it, it's also usually dark. So when I drop the kids in nursery, it's dark, and when I pick them up again, it's dark again.
Oh, goodness. It's like living in Canada. Well, it's not better here in Boston. We're expecting a lot of snow this week. So I thought, scheduling this interview would hopefully get some sunshine into our days and away from the darkness. So for those of us in the, those of our listeners who don't know who you are, can you tell us a little bit about yourself?
Yeah, sure. So my name is Alexander Hauser. I'm a postdoc at the University of Copenhagen at the Department of Drug Design and Pharmacology, Denmark, and I have a part-time affiliation at the Institute for Biological Psychiatry in Roskilde. I am genuinely interested in science across fields, and I'm particularly interested in bridging different domains. So during my PhD in Copenhagen with David Glorian and some time in Cambridge with Madan Babu, I was particularly interested in harvesting and integrating the explosion of scientific data from different fields, such as structural biology, evolutionary biology, with its sequencing of various near and distant species, cell biology, and pharmacology to gain new insights into GPCR biology. And there have been three main areas I was working on, one being a new coevolution, machine learning, and rational approach to tackle orphan receptors. So to find new endogenous peptide ligands that bind putative peptide orphan receptors through learning and understanding of currently known peptide hormones and their cognate receptors. And another one being pharmacogenomics of GPCR drug targets. So trying to grasp the first idea on the spectrum and prevalence of human genetic variations in GPCRs and the impact on health, disease, and drug response, given that GPCRs are such a working horse in drug discovery. So there we identified that variants are widespread and continue to arise and likely contribute to adverse reactions through various different mechanisms. So for my postdoc now, I decided to continue that path and work on the integration of different fields, being genetics, molecular pharmacology, and pharmacoepidemiology and psychiatry, which we now refer to as precision psychiatry. So in order to do this, we collaborate with a big Danish biobank called ISAC, which constitutes of 100,000 psychiatric patients, which have been genotyped and, more importantly, can be linked to other healthcare registries, such as prescription registries. So this gives us the opportunity for the first time to link and study people's GPCR mutations in the context of psychiatry and individual drug treatment, which is, I think, it's very exciting. So although I'm still at the same department where I did my PhD, and where I mostly interact with pharmacologists and computational chemists, I'm now talking to geneticists and neuroscientists and psychiatrists, experts in pharmacovigilance and pharmacoepidemiology or big data, where I really learn a lot and which is very exciting now.
Wow,
so
that's
a brief summary of what I've been doing and what I'm sort of continue to work on.
That's really phenomenal because you get to talk to and work with so many disciplines, and yet still you're focusing on GPCRs, but at a whole different dimension, which is phenomenal.
Yeah, exactly. I'm trying to stay in the GPCR field while trying to bring in new methods from other fields and expanding on sort of the clinical implementation and investigation as much as possible.
And having access to the data from patients, I think, is just phenomenal. It just gives you such an amount of resource. And I think every time when human patients are in the mix of a research topic or research study, it is very hard to get access to these patients. It's very hard to get access to the information about these patients. And you're in this ideal position to work on GPCRs, but also have the clinical data.
Yeah, true. And Denmark is really unique in that sense because here everything is registered through the CPR number. So that's kind of like a taxation number. Mm-hmm. But basically everything is linked through this number, from grades in the school to your disease history to where you've been living and your income. So that's very unique, and that doesn't exist in many other countries. So you can actually do these nationwide studies, whereas in other countries you rely on having specific cohorts where you select people to be part of your biobank, for instance, such as like the UK biobank, which is a somewhat biased and skewed population and doesn't reflect the entire population of a given country. So that's why it's ideal being in Denmark, although it is still very difficult to actually gain access to the data. And obviously it's not just going online and asking your query. I mean, you have to write applications and you have to be granted access, and then it can still be difficult to actually work with the data because it lays on some statistics Denmark service, which are some Windows machines, and people may not be used to work with this. And on top of that, then you need to maybe add your own data to it. So you have to work remotely. You can't just pull the data and work with it on your local laptop. So there are still some boundaries, especially now also with GDPR. There's been some issues and problems here in Denmark. So the authorities are a lot more careful, which is good. But it takes time. But it's very exciting, and I'm looking forward to this, to really fully embarking on this project even more in the coming months and years.
Wow, that's fantastic. And I'm glad that it's hard to get access to these because it is protected data. It is personal data. And so for those of you who don't know, so the GDPR is actually these laws in Europe that are, I guess in Denmark as well, that are to protect people's identities and personal data and information. And I'm glad it's happening, but I'm still also happy that, you although you have a lot of hoops to jump through, you still can do valuable research and valuable work with using this very important data.
Yeah, and these people haven't given consent. So it's different to, again, like the UK Biobank, where people have given active consent for their data being researched. Whereas now it's the people in Denmark, it's the people who are going to the pharmacies and filling a prescription. They don't consented me to accessing their data. So that's why we have to be particularly careful. And then, of course, we're looking at the genotypes and linking it through various other data sets. So, yeah, it's good that we have to be very careful and have to go through a lot of training that, no issues arise later on.
Of course, protecting the integrity of the data, and also protecting the people who actually offer without their consent the access to that data. Before we continue, I wanted to ask you, when did you first get interested in science? Do you remember?
Yeah, I think I was always interested in science. I was never sure that I wasn't like this person who knew, okay, I'm gonna pursue a scientific career. I want to be a scientist. I was always interested in biology and physics and medical sciences. So before actually finishing high school, or just about the time, I was doing an internship at a hospital to figure out whether I wanted to do pursuing a medical career. But then I realized that it's actually not scientific enough for me, and it actually gets lost in the day-to-day practice with patients. So then I decided to study a very broad science background.
That's interesting. And how old were you when you made that realization that you're more interested in more of the scientific and research aspect of science?
Yeah, that was so after high school, I left to travel in Australia for a year, doing one of these working life work what was it called? Working visas in Australia. So working my way around the country and learning about life basically. And I really enjoyed that. But then, of course, I had to make a decision what to do once I'm back home. And I think then I started also applying for universities, and I realized, okay, it has to be science one way or the other. I still didn't know where exactly I'm gonna go, but something with a broad, flexible undergraduate studies I thought would prepare me for sort of the decisions to come, where specifically which niche to focus on eventually.
That's interesting, because the broad aspect is still there in your current day, where yes, you're working on GPCRs, and that's really narrowing it down, but you're using this idea of looking broadly at a field even today.
Yeah, that is true. Yeah, maybe that is a reflection of my history that I
Yeah,
haven't
I think
fully decided even yet at this point.
it's a reflection of, I think, of your personality, which is great, because some scientists, even in the field or outside the field, choose to focus on one protein for their entire career, and others choose to focus on a broader aspect of a topic. And you're using, not only you're looking at it broadly, but also you're using all these different tools to kind of understand the field. How did you get into computational work and machine learning?
Yeah, so maybe I just say, like, I'm actually German and started my studies in Münster in Germany. And as I said before, my study program was fairly flexible, which allowed me to do lab rotations and internships and exchanges abroad, which I really liked. So during my bachelor's, I was actually doing some work on mycins using turf microscopy. So I have this experimental background.
Mhm.
Then I applied for an exchange program in India, in Hyderabad, where I was in doubt whether an experimental project would actually be feasible and very, very wise to do. So I looked into computational groups to do my rotation and my exchange. So that was my first exposure to computational chemistry. So
Oh,
with very little resources, I was doing 3D QSR and modeling and virtual screening on a bacterial protein. And then coming back to Germany, I was looking for more computational projects because I really enjoyed doing it, which allowed me to do an internship at a small company called European Screening Port, where I continued computational chemistry projects on peptide and benchmark docking results for peptide docking. The company then transformed into a Fraunhofer Institute, and I was intrigued to do a PhD there. But my fiance was actually keen on a master project in Copenhagen, so I looked around for computational chemistry groups in Copenhagen and Denmark, and started a master project with David Gloriam.
that is so interesting. So how many countries have you lived in or visited during your training. I didn't realize that you were also you spent time in India.
Yeah, I really enjoyed traveling, and as I said, like my study program was actually quite flexible, and that was really great, and I really made use of it. So I've had besides this half year in India, which we combined with some interesting traveling. I spent some time in Sao Paulo in Brazil for a malaria project. Also, still under that, I think that was doing my master's. And then I came to Copenhagen, and then as part of my PhD, I've done an external stay in Cambridge with Madame Babu at the LMB. And then later on, when my partner did an external stay in Boston, then I followed her basically as part of my parental leave. So spent some time over there as well.
Well, true scientist and a true world traveler. I think it also goes back to the openness and to the breadth of experiences that you have. And when was the moment where you kind of encountered GPCRs in general?
Yeah, that was when I started joining David's group. That was really, I haven't really been exposed to GPCRs before, other than undergraduate training, but that wasn't where I particularly picked up my interest. It really came from looking for a computational chemistry group, and then David was there, and he's had a long history of working with GPCRs. So I started with him, and then he offered me a project working on the deorphanization potentially of orphan receptors, and that's basically where I started working with GPCRs and working with GPCRs ever since.
That's awesome. So, since most of our audience is more bench scientists or people who are more pharmacologists or molecular pharmacologists, can you give us an example of a typical day that you have nowadays? Well, now we're in a COVID situation, but let's put COVID aside. How does your day look like? And for example, how does your week look like usually in the lab or in the office?
Right. Yeah. Interesting that you're saying that most of your audience is experimental in nature. Maybe you've done a survey, but maybe there are actually quite a few computational people listening to this as well. But yeah, so yeah, I think that my typical day or week have really changed in the past. So it really depends on the project. So there isn't really like this one method that I'm going through again and again. I've made a little bit of a transition from computational chemistry, or quite a big of a transition, to more classical bioinformatics to computational biology. So as I said before now, what I said before, working on this precision psychiatry project, it's really more about data integration and combination of methods that are more based in the genetics. So going through the genotype data, going through exome data, doing quality controls on those, selecting individuals, and then doing pharmacoepidemiology work where I'm looking for people's prescriptions, and there's a lot of confounding statistics that you have to do to select people based on their age, and not to have any bias in your data. But then I still have projects where I'm also doing structural work, so taking data from GPCR and passing it through evolutionary biology methods, looking at their structures, looking at interactions between ligands and receptors, using different programs like Pymol or Maestro. Then, I'm mostly scripting. I'm mostly programming using Python, making visualizations using different packages, and doing statistical analysis using different packages within Python. So it's very different, and it has various stages depending on the project. So it's really, there's no day is the same, and that's also what I love about science in my position now, that every day is different, and every day I'm learning something new.
Which is phenomenal. Yeah. So you're used to handling large data sets and you're used to manipulating large data sets, which someone who doesn't have the skills and the experience of using such techniques and such software packages, it might seem very out there, very UFO type. But then again, I think it also again reflects your ability to use so many different tools in order to answer these questions. So you had mentioned that you were in Boston while on paternity leave, and being a postdoc, I think at least in the US, not all postdocs get to the point where they can have a family and they have this, this work-life balance. Well, we all struggle with what you'd call a work-life balance, although I feel like work and life should be the same, because you should be the same person going to work and being the same person outside of work. But can you tell us a little bit more about how you manage, having a family and at the same time doing the work that you do?
Yeah, so one reason to stay in Denmark was because, of course, we liked Denmark and we both had opportunities to continue research as PhD fellows. But we also very much like Denmark for its child-friendly culture and environment, because we were pretty sure that we wanted to have children early and during our PhDs. But this would have been a magnitude more difficult in most countries like Germany, where you are not so financially independent as a PhD student. It's also very uncommon to have children in most countries other than the Nordic to have children while pursuing a doctoral degree. And Denmark has very good parental leave conditions for both mother and father. So we had the opportunity, or my partner had the opportunity to join me going to Cambridge for half a year while being on parental leave. And then 3 years later, I had the opportunity to take extra parental leave to join my partner when she was doing a project at the Broad Institute. So that, I think, was just a great opportunity to still continue on the academic research while also maintaining a family life and being very flexible. I mean, me particularly, because I can work and catch up on work during evenings, and I'm more flexible than my partner, who's still doing experimental work in the laboratory. And being very flexible as an academic helps a lot in maintaining the family life. So we have now 2 girls, one and a half and a 4.5-year-old. And I think we can say that we're very, very, very happy that we prioritized having children early and doing our PhDs while staying active in research.
And I think it's a unique situation. I know one person who had both of their children during their PhD, but I think also it was at Rockefeller University, and it was a very great environment for that because you lived on campus, you had childcare opportunities on campus, and that was really, really easy. And I've heard of people who had their children, I think also during their PhD, but then they decided to move back to Rockefeller because they would spend hours on the road dropping off the child to childcare and then going to the lab. And so it's definitely not a conductive place, at least here in the US, or even in I think a majority of the countries. So Denmark seems to be that very friendly for that.
Yeah, and it helps if it's very normal. Like if your colleagues having children and your bosses have had children early,
yeah,
then there's no shaming in going home early, picking up your children at like 3:30 in the afternoon,
yeah,
when most people are doing it anyways. Well, it's a different working culture both in the UK and in the US, where if you're the only one, then you always feel like you're not catching up, you're not doing enough,
yeah.
and you always have to justify why you're now leaving early. And of course, it's not just leaving early because you don't want to have your children in daycare all day, but it's also they get sick very often, especially in the first year. So you have to take time off, and then of course in the afternoon you spend time with them, which is great. But then other people are able to work and you are not, and then you have to catch up whenever it's possible. So having a culture around it helps a lot. And then of course having the allowance, both financially, but also by the government and by the university to take actually substantial amount of time off to focus on your family life. Like I actually just came back from 7 month parental leave this year and coming back in September, which was ideal with the pandemic because then I felt at least it was a bit easier for us than maybe for some others, and we were really able to enjoy the time together and had the time to really focus on the family aspects, while other people maybe didn't work on their projects, and then I mean, you're never really totally off the grid, even though you're doing parental leave. I mean, there's always emails coming in and so on. But I feel like this time now, during my second parental leave, I had a little bit more time to focus on that, on my actual parental leave, because also in the lab was less activities. And now I even had the chance to still, I mean, when the little one was sleeping, to zoom in on some of the meetings or some of the group meetings and talks, whereas before everything would have been at the university and I would never had the chance to still follow some of the interesting things going on at the department. While this time it was a good mix.
It's so interesting that you say that, because I think I can very much relate to the fact that, yes, being on parental leave doesn't mean that you're completely disconnected from the science, that you're completely, in your parental leave bubble. I remember, taking time off when my son was born, but 2 weeks after he was born, I was running samples at the flow core facility because I was curious about the data. And it was in the middle of the night, but I still went. It wasn't publication quality, but at least it gave me an idea on the differences between the different samples. And I just could not let the samples, end up in the trash just because I was on maternity leave. And I think a lot of us scientists have this interest in getting answers to those questions and still remain connected to the science, which I think is pretty unique.
Yeah, people always say there's never the right timing having children. And I'm just imagining now, looking forward, now that I'm more actively supervising people and I'm more looking into applying for my own funding, establishing a group, and the same applies for my partner, who is now the opportunity to establish her own portfolio and her own group. Like going forward, it would be much more difficult now, taking time off and taking long time off, focusing on children than already having the children, and now them growing older and becoming more independent. So also, like, there's never the right timing, but I feel like it really, like, going forward, it helped that sort of we could at least tick off some of those boxes. And I mean, while of course it is just a pleasure coming home, and it distracts also from your worries and your long to-do lists and all your projects and ideas. If you just come home and you play with your children, and they make you happy and they make you think about other things than your science, and then it's really refreshing. And then once they're in bed, then I have a fresh mind to go back to my projects and my ideas.
How is it living as a German national, living in Denmark and living, in English, but also in a Danish society?
The Danish society is great, and it's very easy to arrive and to live in Denmark, because everyone speaks English almost as their first language. I mean, it's not their first language, but I mean, even the bus drivers and people that you usually think, okay, in other countries, maybe, they don't speak English. Everyone does in Denmark. So for the short term, it's very easy to arrive as a foreigner. And there is a big international bubble in Copenhagen. It has a very international focus, very international touch. So it's a great country to live as an expat. But going more long term, it's a bit more nuanced, especially now for us having to make a commitment to stay in the country or moving somewhere else or moving back to Germany. Then you're starting to feel like, okay, there's some things I'm actually missing from my home country. And I mean, of course, the language is one thing. Given that it is so easy to speak English in Denmark, you're actually never exposed. You never feel the need to learn and catch up on Danish, even though we've done Danish courses in the beginning. Like if you don't really train it, then you also don't get any better. So I mean, obviously at the university we speak English, and then given that we both German at home we speak German, so there's very little exposure to Danish. But now our children, I mean, they're in daycare speaking Danish all day. So at some point their Danish is going to be better than our Danish, and maybe it is already that for the older one that she starts singing all the Danish Christmas songs, and I don't know the lyrics. And then she complains like, "Oh, you don't know the lyrics." And then, yeah, I know them in German or in English. Let's sing them together. But then she doesn't know them. And so it feels like sometimes it's a bit of a shame that there is yet another aspect that you have to take care of, and it's certainly not as easy as it would be in your home country. I mean, it's really just like these like small points, percentage points that. If I were to be, fluent and native in Danish, then it's it's one less point to think about. Like, it's one aspect now being the Christmas songs, another one being like you have to catch up on traditions and what you do in a certain situation, and you have to learn all that, and that's great. You learn so much every day, but you always have to be a bit more concentrated and conscious about these things, compared to a native in the country.
Wow. You had mentioned that so now you're mentoring more people. What type of a mentor are you? How do you approach mentoring?
You mean like being mentored, or I'm like
in mentoring others in the lab? Now you're a postdoc, you're in a more senior position, you're looking to establish your own team. How do you approach mentoring?
Well, first of all, I try to lead them on the path not to make the same mistakes as I did. I mean, these can be small things like when it comes to coding, for instance, to really start thinking through how to do things from scratch, and provide all the tools that took me sometimes years to establish to be productive. So that's very practical, and I think that's very general. Also, whether or not this is computational or whether this is experimental, you basically teach your experience, and I think that's sort of my approach. But then also having a constant trying to have a good relationship with the people, and I mean both of course on a work, focusing on work, but also privately, to at least to test out whether one is on the same wavelength also personally, because I think that helps. And I've been very fortunate to have been very close to the people who mentored me and are still mentoring me along my way. So I think it helps if both sides get something out of such a relationship, and if both sense that the relationship is earnest and it's not just because you happen to be that supervisor.
Yeah,
it's very important that you feel that it's a mutual relationship, and you learn from each other. Because then I think it's also important that it continues even after that person has left. Or I mean, same for me when I moved on, and then it's very easy to move on and be distracted with the new projects and ideas, which can make it difficult to keep up those relationships. So I'm hence trying to remind myself of people that are important to me and try to reach out to them occasionally and hear how they are doing. I mean, not just with scientific mentors, but also friendships that I started along my international journey, which can be sometimes difficult given the physical distance and busy schedules. But I think it's important to develop a relationship.
I absolutely agree on the fact that, yes, so you need to the goal of mentoring someone is actually showing that person what not to do or what the mistakes that you made, and it's by letting them observe what you do, but also having that personal relationship and that caring relationship, that you're there to support each other, and you're there to you know have a great foundation of that relationship and build upon that, is really crucial in the long run, both on work, quote unquote work relationship, also as a personal relationship. So one thing I like to do is, when you were talking about maintaining relationships and sending out messages and keeping in touch, is that in my Google Calendar, every time I learn somebody's birthday or somebody's kid's birthday, it goes into my Google Calendar, and I always send out a short note, hey, thought of you, I know it's your birthday or it's your son's or daughter's birthday, happy birthday, hope everything is well. And I think those little attentions are the ones that change that relationship from a, for example, work relationship to a really personal, at the end, meaningful and deep relationship.
Yeah, I agree. I do the same, and I'm trying to do the same also with like former master students, for instance. I supervised whom I know doing or pursuing a PhD, even though that sometimes is in very different fields. I still try to catch up every now and then and hear how things are going, even though it's a completely different project, and there's probably no overlap or future overlap. I mean, you never know, but I think it's still. I mean, first of all, I'm genuinely interested in what people are doing that I supervised, and of course I hope for the best for them and what they are doing, and maybe I can even learn something along the way of their science, because I know that we probably had established some sort of similar way of thinking. So for me, learning from them is maybe easier than learning from just reading a paper, or maybe I wouldn't find the interest in reading the paper, but now I have a sort of a connection to it, and then I know, okay, I know a person in this field, and it kind of feels good to know where people are heading. And yeah, as you said, like at some point maybe people, I mean, paths can come together, of even though it's maybe a totally different area.
Yeah.
But since my interest is also to bridge areas and bring different areas together, I never know whether these people are not going to become valuable again also to me.
You touch on a very valuable point, which is collaboration, but also on networking. And I think networking and the knowing people and showing an interest in what others are doing and keeping your network as wide as possible is also very important, not only for your self-benefit, but also from a scientific perspective. I think doing science in a bubble is not something that is productive in the long run. So if there was a take-home message from these last couple of minutes that we've been talking, is that reaching out to people, talking to people, networking with people, caring about the person, but also about the science that they're doing, is just the best way to increase your visibility, but also maybe contribute. You never know who might need your help at some point to do something, and then where a collaboration and a great success comes out of.
Exactly. Yeah.
And then reaching out, sending an email. I've had so many discussions by now with a lot of my podcast guests, and I was happily surprised, and that everyone on the podcast said, you read my paper, you want to talk to me, just shoot me an email, and if I have time, I'll chat with you. And this is a message to send out there to everyone who's listening to the podcast. It's not as scary as you'd think to just reach out to people and just ask for 5 minutes and have that conversation, because you never know where those conversations lead. And I think most of the most successful collaborations and work that has been published, not only in the GPCR field, comes out of just that chat over coffee at a conference, or meeting people, and
Yeah,
those kind of.
It's a bit of a shame now with conferences all going online, which has some advantages, but I'm also very much in favor of this hallway track at conferences, and
Yeah.
I really like to just bump into people that maybe I haven't met before, and start talking to them and hear what they are doing and to learn from each other. And maybe there is something in for both of us. And especially me working with data sets, of course, it's a bit easier for me to establish new collaborations and integrating and start working with other people's data than maybe for other people who are more focused on specific experimental technique or working on a specific target. So that's also why it is sort of becomes more natural to me to be interested in other people's sciences than maybe for other people.
Yeah, I think it all depends on kind of your nature, and I think as you mentioned, you do have that expertise where basically, in theory, and please correct me if I'm wrong, you could be collaborating with a lot of people because you have the expertise to manipulate large data sets. And it's like, the way I see it, it's like this big box, and no matter what you put in it, as long as it's in that box in your realm, then you can just manipulate it and ask the right questions when it comes to manipulating data sets.
Yeah, it's true. But I think it's not enough to just being able to manipulate data sets, and many people are able to do this. But you really need to also combine it with domain knowledge.
Yeah, that's true.
Certainly wouldn't be easy for me to, just apply the same to, other protein families or, other areas, because there I don't have the domain knowledge. Of course, you can of course read up on those, but it still takes time to really grasp what are the big questions and what are the bottlenecks. And it's also very important to know how the data has been generated, because otherwise it's just numbers and labels, and then it becomes very, very quickly very, very meaningless. You need to know how the data had been generated and what are the caveats of the data. What can you interpret from based on these experiments and whatnot. And so it's very important for me to understand the experiments, and if I don't understand, then to go back to the people who created the data sets to really know where are the limitations combining it, for instance, with other data or interpreting the data from a different perspective.
I think that's a fair point that you're making, but I think in that sense, having the right collaborators who can guide you on that, and you can learn directly from them, from these conversations that you were mentioning from different fields, for example, that's when these relationships become valuable. And you get to a point where, yes, you have the ability to manipulate virtually any type of data set, but having the right relationships and learning from people who are experts, because you can't be an expert at everything, but experts in a specific field, like you are about GPCRs, I think it's just, that's the beauty of collaboration.
Yeah. And sometimes it's just about knowing the experts and bringing the experts together at the same table.
Exactly.
It can be obvious now, with the people in the GPCR field, because people know each other actually quite well, and I feel it's a tight-knit community. But as I mentioned before, that now I'm working with geneticists, and I'm working people experts in pharmacoepidemiology and drug safety, and now I'm coming as a GPCR molecular data integration scientist. They don't know anything about what I'm doing and what the GPCR field has been doing and how that relates to their fields.
Yep.
And they don't know sometimes about each other, or most often they don't. But I feel like there is similar interest in the same long-term goal from different perspectives, and sometimes it just requires someone to bring those people together to create synergy and new insights into the sort of a common goal and common question.
I could not agree more. You had mentioned that you're working on building up your own group and getting your own funding. Would you want to continue in the same type of working on the same type of topics with this wide variety of experts to answer common questions as a group leader?
Yeah. So given that I only sort of recently fully embarked on this precision psychiatry field and topic, I think I'm gonna stay with the personalized medicine and pharmacogenomics for the years to come, and then we'll see where the journey is going. I have a lot of other ideas and interesting scientific projects, and I just need some people to help me out on those and to do some preliminary analysis on those. And if something comes up on those, maybe this is something I could pursue as well. But for now, I'm very happy where I am, and with the pharmacogenomics in Denmark and all its advantages with the data and the people that I've identified here with similar interests in psychiatry and bridging the different fields. I think that's very exciting, and Denmark has a lot of funding opportunity for people like me starting their own potential groups. So that's probably the path forward.
And you're working on a set of very hot topics as well, which will have a deep impact on drug discovery in the long term. I think precision medicine is such an interesting and important topic. We don't react the same way to the same drugs, and we need to understand how to specifically, design drugs to specific people at the end of the day, when you think about it. And I think it's just a phenomenal way to attack your next step in your career.
Yeah, thanks. It's not always easy to convince the clinicians with this. It sounds always great, but then when you actually have to prescribe a different medicine, I feel like, not having talked to numerous clinicians, there's still some hurdles to take, and we need some more proof that this is actually going to be valuable for the individual patient. So I hope I can make some contributions.
I'm sure you will, and I think clinicians are. And it's a little bit kind of, that's what you were mentioning, is that when you did your internship in a hospital and you realized that actually that was but it was too routine for you, and you were seeking more excitement. And I think at some point clinicians, although very valuable, don't get me wrong, get lost into those because they have values, they have thresholds that have been established. And again, it's that. It is what it is, and not everyone has that openness as a clinician to think about the vastness of the data that's there and the genomic variations between patients. And just because these standards work for patient number 1, it doesn't mean that they're going to work for patient number 4.
Yeah, that's also why I think it's important to have them on board very early on.
Yes,
because otherwise you just throw another paper out there, but then it doesn't get traction because it stays in the community, and which can have valuable impact in the community. But if your ambition is that it has an impact on the patient, then you have to work with the people who are eventually using that information.
Exactly.
And it's very important that they have their input and their saying as well.
Exactly, because they're the ones who actually deal with the patients. And I think it's I've just recently experienced this frustration about clinicians or doctors having in their mind, depending on which doctor you talk to, which specialist you talk to, for the same test they had different reference values. And I thought, as a scientist, it just did not make sense. Like your MDs, you have different specializations, but come on, it's the same patient, it's the same test. Why would you have different standards?
Right.
Depending on that. And I think as scientists, that's where we need to provide more data, provide more insight, and better understand the variations in the human population to show that yes, you need to have some kind of a standard value to specific tests or to specific reactions to drugs, but please, let's just find the right value that kind of makes a little bit more sense and not have, depending on the specialty, different references.
Yeah.
For the same human.
Yeah.
Which I thought was really frustrating for me as a scientist, and I kept asking, so why is this value the way it is, and who established this, and how come you have different standards? Which again, I think with your work, better understanding precision medicine and better understanding how to take into consideration the genetic variations in the population is absolutely valuable.
Yeah.
So let's roll into the last of my questions. So you're a postdoc now. You're getting ready to run your own group. What would be your advice to junior scientists, to master students, or PhD students who would want to, embark on a scientific career, but at the same time contribute to the GPCR field?
Oh, to the GPCR field specifically?
Or
Well,
any field.
more generally, I would say go where your heart is and where you really feel engaged in the project. I think that's very important, rather than just selecting what is hot topic, or just going for the big groups or big metrics. Try to really listen to yourself and like, what is it that keeps you motivated? Because nothing beats your intrinsic motivation. If you're interested in a certain topic, then you're going to find the experts, and you're going to find a way to make it an interesting project. So that advice would go for master students who are looking for a PhD project, for instance. Like, now you've had 3 or 5 years of education. What is it that you're really, really interested in? So that would be one advice. And then, I mean, for the GPCR field specifically, of course, that really depends on the person then, and what that person is capable of doing. I think that, I mean, like, more generally, I think the GPCR field would maybe need some people who are interested in going, understanding the pathways and their downstream effects a bit better, for applying phosphoproteomics or transcriptomics to really grasp an idea of what's going on downstream. So having more people on that would be great, I think. But you need the right people to do so.
And to train with, you need to find the right mentors that are doing the science that you're interested in, but at the same time with whom you have that affinity and that ability to work together well, on a scientific but also on a personal level.
Yeah, find someone that resonates with you, and I think then you can actually achieve a lot in a very short time.
Agreed. We didn't talk too much about science, but this is a question that I ask all of my guests. Do you have any aha or eureka moments that you had as a scientist during your trajectory so far? Any discoveries that you were either surprised by the result, or it was something really out of the box, you expected it, but finally you got the data and you said, I'm happy, this is exactly what I was hoping for?
Yeah. So what I mentioned, my first project was finding new peptide ligands for putative orphan peptide receptors. So I designed this library largely based on knowledge on known peptide hormones.
Mm-hmm.
And then I've been going on. So we synthesized 218 peptides, and then we sent it to our collaborators in-house in our institute to test on various orphan receptor cell lines. And then I had time to embark on other projects, and I've been on parental leave, and I've been abroad while our collaborators been working on the data. And of course, we've been in close contact, and we met, often during lunch. And I was always very interested in hearing, what are the first results. And when I first heard results of some initial hits on some of these peptides, I was just very happy that it worked, and we have some results here. And of course, like, you never have that that moment where at least not in pharmacology and such a screening experiment. You're always in doubt whether is this really something. Like, we have a hit, but we have to do some orthogonal experiments, and we have to do those response curves, and every new result leads you to be more hopeful, or be more be happy about the outcome. But it's never like from today to tomorrow that all of a sudden there is this. I mean, maybe for some in some moments there is this discovery moment,
Yeah.
but I feel in science you always have to be skeptical about your results. So you can, every time you have new results, you're less skeptical, and that can make you happy.
I think so too. I think just getting that answer for a hit is already a super feeling, but then getting that hit just opens up the door for more questions, for more
Yeah.
experiments, because you absolutely need to confirm if that hit is really a true hit. And if it is, the next question is, okay, and what does this mean? Or how does
Yeah.
this affect the field, or how does this affect the receptor?
Yeah.
But still, I think the feeling of getting, out of 200 peptides, getting just 10% of those being a hit is just magnificent already.
Yeah. Just the feeling, being able to raise more questions, that is already a great achievement, I think.
I agree. With this, I'd like to thank you, Alex, for your time. I hope you had a good time. I did. I certainly did. We learned a lot about you, and we learned certainly a lot about Denmark. I'm pretty sure you're going to get emails from people asking you. So tell us more about your research.
Sure.
And with this,
thanks. Was a pleasure talking to you and talking about these aspects. And
thank you so much.
If people are interested, then of course they're more than welcome to reach out. Let it be about Denmark, but even more so about about my research, or please reach out if there is anything.
Absolutely. On the website page that will be dedicated to this episode, people will find your contact information. You're on Twitter as well, and very active on Twitter. So if anyone has questions about your research before, or in the meantime, they can definitely find you on Twitter. And also, you're pretty searchable. You have a great publication record, so I'm pretty sure a lot of people know who you are.
Thanks, Yamina. Really appreciate your kind words, and just for establishing this new concept, Dr. GPCR, and the podcast, and the whole umbrella network and environment that you're creating. I think it's really, really great and really useful for the community.
Thank you so much, Alex. I really appreciate it. And with this, take care and talk to you soon.
Bye.
Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest and our Dr. GPCR team, namely Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. A huge thank you to our Dr. GPCR Ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem, so don't forget to join us today. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Soon, you'll be able to share the Dr. GPCR University. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.